Neuroplasticity is a beautiful concept. Now, here people say that a child only develops during the initial years of his age. And once the brain develops till the age of 5, after that, our brains remains the same. So it is the skills and abilities or whatever we learn. It is only during the initial years that we learn well. And later on, our brain more or less, it stays the same. But that is not the case. Neuroplasticity has completely changed the way we look at the world, the way we look at the human brain. So neuroplasticity exactly is what? It is the ability of the brain to keep changing throughout the course of life. So these changes occur in the brain by what? By making and breaking of the synaptic connections between the neurons. So our whole brain is full of neural connections. There are many neurons in the brain. Millions and billions of neurons are there. Since we were born till now, there have been, a lot of neurons, and they keep on breaking and making, and it depends on what we are doing in life, what decisions are we making. It's the genetic makeup, it is the structure of the brain, it is the environment. There are multiple factors that influence the making and breaking of synapses. So that keeps occurring throughout the life. Neuroplasticity, the neural network that is there in the brain, it completely changes the brain, and these changes can be due to genetic or due to environmental. So before we go into the technicalities of what are the terminologies in neuroplasticity, what you have to write in your answer when the exam comes, before going to that, let me explain neuroplasticity in a very, very layman language so that you understand the concept really well. So let's say this is your brain, and there are several neural connections in your brain. Throughout the brain, there are millions and billions of neural connections that are there, and there are synaptic connections of the neurons that are there. Now in this, what happens is there is a certain connections that have been made already. So let's say I have certain kind of connections in my brain, and I all of a sudden I feel like, okay, let me just learn dance. It's a new skill. I've always wanted to learn dance, but I never got a chance to do it. So let me do it now. And I join this class and I start learning. So initially I'm performing very poorly, and gradually over a period of time, maybe like say 10 months or so, I start to perform better. I'm mastering the skill, and my abilities are improving in dance. So what is happening? Whatever the functions are needed for dancing, your motor skills, your balancing, your movements of the body, all these things, the muscles and everything. So all these things that are needed for the dance, those specific connections in the brain start to develop. So there are more synaptic connections that are getting made in my brain for these certain things. Or for example, let's say I had never driven in my life, and I always used to take a cab, or my family used to drive for me or something like that. And all of a sudden I think now I need to work on my driving skills. I'll start learning how to drive. So when I start learning to drive, when I'm driving, I am using so many sensations. I'm using so many stimuli which I was never using before this. So for example, I am able to map the city. I'm able to understand the maps of the city. I'm able to know there where this road is leading to, how to see that the car is coming from the right side or the left side, or any car is behind me, how to do the reverse, and all these things about the driving. So I get to understand all these things, and I start paying attention to all these things which I had never done before. So what happens? Whatever is needed, whatever skills are needed, whatever connections or areas in the brain that is being used, in those particular areas, the synaptic connections start to build up stronger. So more I drive, better I get, because more synapses are being connected. So those specific areas, the spatial ability, my motor movements, and my attention to all these things, those particular parts of the brain which are being used to do that ability, those parts of the brain start to get developed. So basically in neuroplasticity, what happens? If you are using your brain in a certain thing, those synapses are going to become stronger, and the brain parts which you are not using, those began to break. So it simply means that brain thinks, okay, you are using your certain parts of the brain for this thing, I am going to make more synaptic nerves for this very thing, and you become better at it. If you're not using your brain thinks, okay, maybe this part is not needed anymore, so there is no point in making more synaptic connections in the neurons for this particular thing, so that part begins to shed. I'll again give you an example. Now, many times you would have come across, mothers yelling at kids or telling their kids, don't watch TV. Why don't you read a book? Reading a book is better than watching a TV. Why don't you go out to play? Why you keep sitting in a room? And things like that. So all the time we are yelling at our kids to do these things. Now, why do we tell them to switch off the TV? Why do we tell them to read a book? Or why do we tell them to go out and play? The simple reason is, when they are reading the book, they are increasing, the language ability is going to increase, their vocabulary, the familiarity with the words, the spellings, and so on. So when they keep doing it, the particular part of their brain, the specific parts of their brain which are used in reading, your language, your comprehension, your understanding of things, your imagination while you're reading, all these things are being used. When these things are being used, there are more synaptic connections building up in your brain. And when you are not reading, when you are not playing out, when you're not doing such productive activities, what happens? Your brain thinks that these particular synaptic connections are not needed. This person doesn't need all that. So let it be. Let's just, shed these connections. Let's break these connections because they are not in use. I don't do maths at all. I don't do mental calculations. I don't calculate things in my mind, and I always use a calculator. Now, brain gets a signal that this person doesn't need those mental ability skills. So those connections are going to break, and I might, have lesser ability in doing mental maths altogether. But more the maths I do, those particular parts of the brain are going to build up more connections, neural connections. Hence, I will get better in that subject. So that is why when your mother is yelling at you that don't watch TV or don't get into your iPad and mobiles, go out to play, do some productive action, or do some productivity, or read a book, or maybe listen to a nice song, or do some creative art or something, follow your hobby. In that case, please do listen to her, because she is only trying to make or build the neural connections in your brain, and she doesn't want the neural connections to break in your brain. The time we are born till the time we die, these neural connections keep on changing. It all depends, how you use it. I will give you one more example. There are many people who, till the time they're doing their jobs, they are performing so well, they are so active, and they can do a lot many things which are very productive in nature. The moment they get retired, now what is happening? After retirement, they don't use their brain in the way they were using it before. For example, some people, not all I'm saying, some people, they think, okay, now I'm retired, now I don't want to do anything, I'll just sit and relax, so I'll not do anything at all. So what happens is, when they are not doing anything at all and they're just sitting and relaxing, basically the neural connections are breaking in their brain, and they tend to get older, like mentally older, faster. And the people who keep working as in not draining yourself out after the retirement, but keep using their brain, maybe like playing a Sudoku, or doing a gardening, or doing some trading or things like that, maybe reading a book even. So these kind of things, if you keep your brain active, then what happens? You don't get older faster. Why I'm saying older faster, not as in really the aging thing, but older faster as in your brain stays active for a longer period of time, and it stays active in a very good qualitative way. So this is important, that we tell people who get retired to keep doing something and not just sit and relax and give up everything, because you're going to age much faster. So this is what neuroplasticity is. So the first terminology that you need to remember and must write in your answer is synaptic plasticity. What is synaptic plasticity? It is the connections making or breaking at a very simple level. So basically it is the ability of a neuron to make the connection or synaptic connection or break it. So it is at a very simple level on the neuron level. Second important terminology is cortical mapping. Cortical mapping is a larger part. It is larger, on a larger level it is. It means that the brain part X assumes that they have to cover up or take the work of brain Y part. So this basically means maybe, during the time of injury or something, or brain damage or something. So what cortical remapping really means is that when a person, for example, there is a person who meets with an accident, and when they met with an accident, they got a head injury and some part of their brain got, hurt and damaged. So in that, what happens, let's say, for example, this person lost the vision. So now what is happening? The person has lost the vision, they cannot see. Earlier, what was happening? You were seeing things with your eyes, you were listening, you were speaking, and you were smelling. So these are the sensations, and touching as well, of course. So these were the things that were happening. Now, have you heard that people who are blind, they have a very sharp hearing? Now, why does that happen? That is because of the neuroplasticity, cortical remapping. So what happens is, let's say there is a person who just met with an accident and lost his vision due to head injury. Now, when the person lost the vision, earlier, let's say he was using his, of course, eyes to see things, and then, ears to hear things. But when the vision was lost, now the sensation that eyes were capturing earlier, now that is not happening. And all the load of eyes are now have fallen onto the ears. So now ears have to take up that place and see that I need to sense things around me, sense the environment around me, and all the load is on me only. I mean, my ears I'm talking about. So now because eyes are not there, I cannot see things, and number of things I won't get to know. So my touching, my smelling, and my hearing, all these sensations sharpen altogether because I'm using it more to sense my environment around. So earlier I was seeing with my eyes as well. So ears did not have much load, or smell or touching was not having much load. They all had, kind of load was all distributed among all these senses. But now when the eyes are not there, what has happened? The load has increased on the other senses. Now they are responsible for understanding that what is happening around the environment. So if you see a blind person, their smelling and their hearing is more sharp sometimes than the normal person, because they have to use their ears for understanding what is going on in their environment, or smell or touch to understand, because the eyes are not there. So that is why when they are using those parts of the brain more than before, the synaptic connections in those particular areas have now deepened. They have now become more strong than before, and now that is the reason their hearing and their touching and smelling has increased as compared to before. Somewhat cortical remapping. Another example would be like, for let's say somebody loses one of their arms, and let's say it is the right arm. Most of the people are right-handed, so I'm just giving an example here for the right hand. So there is this person who met with an accident, and due to some unfortunate circumstances, that person, what happens is, earlier the person was doing lot many tasks only with the right hand. So the right hand was the dominant one, and he was using all these things with this right hand. So this was a stronger hand, even like physically, to pull up things, to push and pull and pick up things. For everything, the muscles of the right hand were much stronger for this right-handed person. So after the person loses his right hand, what happens? all the load has now come to the left hand. Now the brain knows that everything that is to be done has to be done by the left hand. There is no other way. The person doesn't have right hand now. So what happens? The person starts using his left hand for eating, pushing, pulling, picking things, and so on. So when the person is using his left arm, earlier which he was using right arm for pulling things, and now right arm is not working, now they have started using their left hand, left arm, the muscles of the left arm become much stronger. Why? Because they are being used more. So anything that is being used more becomes more stronger and better. Anything that is used less becomes less stronger. So here what has happened, this switch of the muscles and usage has happened. There's this beautiful saying in neuroplasticity, I really like this, it rhymes really well. Neurons that fire together wire together. There's this beautiful saying in neuroplasticity, I really like this, it rhymes really well. Neurons that so this simply means that neurons that keeps firing as in communication is happening continuously. So the usage, it's being used basically. So when it keeps firing, the neurons, those always wire together. So neurons that fire together wire together. Neurons that fire out of sync fail to link. So that means the neurons which are not able to sync, they are not able to link. So it is good. Please keep using your brain for all the productive activities that are there. So that means the neurons which are not able to. Another is axonal sprouting. Even that is very interesting. It means that every axon that starts does not mean it will reach up till the age of the full functioning of axon. Basically, sprouting is what? Sprouting is early growth of an axon that may or may not complete the journey of reaching the target neuron and restoring function. So it is the key structural change that makes the plasticity happen to facilitate development of normal neurons and recovery of functions of injured neurons. Sprouting declines with age. Axonal sprouting is a process where fine nerve processes, sprouts, grow out from the intact axons to re-innervate denervated muscle fibers. Thereby, the sprouting sustains the nerve supply to muscles and in turn the ability to move. So what is happening is that when the axonal sprouting happens, sprouting is what? Just starting of the axon. So when the starting of the axon starts to happen, it does not mean that it will be fully functioning. Only if we use it, it is going to grow and have further dendrites like branches kind of a thing. If we do not use that particular area of the brain, we do not use any ability in regarding to that neurons or axons, that axon does not completely grow and is shed off. So when a child is born, there are number of axonal sprouting that is happening. There's so many sprouts, all kind of axonal sprouting is happening. So as the baby grows, like initially in the starting of the lecture, I said that a baby's brain is developed till the age of 5. A child's brain is developed in the age of 5. It does not mean that your brain is not going to change throughout your life after you reach the age of 5. Nothing like that. It keeps changing. But the maximum kind of synaptic connection making and breaking happens during those early 5 years of your childhood. So that means when a child, comes in contact with number of things in their environment. So that is why when we say that, for a smaller child, make a good environment and, present them with good stimulus, positive stimulus. Why we say that? Because we want those particular positive, good synaptic connections to be made and the other ones to be shed. So that is why the reason that we say that, till the age of 5, don't let them watch TV, don't let them watch iPad or mobiles, because they kind of ruin your synaptic making. So better give them some books. Maybe they just turn the pages. They don't know how to read, of course, a 5-year-old. So they might just play with the book or they play with some educational toys from which they are learning. So that is very important. Why? Because these educational toys and books around and positive environment around is going to build up some new good neurons. So what happens, axonal sprouting here is that it only sprouts when it is being used. It reaches up till that age and it makes more connections. When it is not being used, that just gets shed off. So that is what axonal sprouting it is, the early growth of axon, which may or may not complete the journey of reaching the target neuron and restoring function. Then another important terminology is synaptic pruning. Synaptic pruning is the natural process that occurs in the brain between early childhood and adulthood. During synaptic pruning, the brain eliminates extra synapses. Excess of pruning can lead to the onset of schizophrenia. Too little pruning can lead to autism and epilepsy. Now, if you see in this picture, at the age of 36 weeks, that means when the fetus is there, we are talking about fetus here, it's in the mother's womb, only 36 weeks. This is the amount of neural connections that have been made. And the newborn has got these many, a little bit more. 3 months, the neural connections have been increased, and if you see by 6 months and 2 years, there are too many synapses that have been formed. By the age of 4 and 6, if you see, the synapses have decreased. Now what is happening, I'll again explain in a very layman language. Now when the child is born, there are in initial 2 to 3 years, the making of synapses are just too many. There is too many. It's like millions and trillions of synapses are being made. All kind of connections are being made. Now, having too many connections is also bad. It can lead to autism or epilepsy. Why? Because there is too much of traffic jam. There is too much of firing that is happening in the neurons, and which you don't really need. So it is really going to bother a child or a person when too much of firing is happening. So that we do not need. We need only those synapses that we are going to use. So initial 2 to 4 years, whatever the child does, the kind of environment they have, the kind of stimulus that they get in touch with, those particular synapses, brain thinks, now this child, there are too many synapses there, connections are there. Now the brain thinks that we don't need so many connections of synapses. Okay, let's break the ones which a person doesn't need, and we'll keep the ones that a child might need. So this is called pruning. Pruning means shedding off these neural connections that are there. So this shedding of the neural connections are very, very important. Why? Because we do not need so many connections in our brain, because excess or the lack of neural connection both can be a problem. So basically what you have to remember is axonal sprouting was making of it, pruning is breaking of it. So pruning is a very, very important part. Now why? If there is of breakage is there, it can lead to schizophrenia. The person is not able to think right. There are hardly any neural connections in their brain. On the other hand, if the pruning is very less and there are too many connections there, that means a person can have autism or epilepsy because of excess of the neurons and synaptic connections, and there is less pruning that has happened. So in initial, when the child is very, very small, at that time, like 2 to 3 years, that time it is important that the right amount of pruning happens. So we don't want those synaptic connections that will not be using. Only those synaptic connections will be kept in our brain or be kept in our brain, which a child would be needing. So that is what pruning is. We'll be using some technical terminologies here to understand better, and so that you can write in your answers as well. There are two aspects of neuroplasticity. One is dendritic branching and another is pruning. Dendritic branching, like I already told you, the branches are made. Okay, so more synaptic connections are being made. Dendritic branches are like the branches, like just the branches of trees. So these are being, made more and more, and the connections are happening. Pruning is the shedding of the same. Now, one form of neuroplasticity is the strengthening of synapses. Now, when we say neuroplasticity, basically neuroplasticity is both the changing of the brain. It can either be making of the synaptic connections or breaking of the same. So, neuroplasticity is the strengthening of synapses. This is due to the process called long-term potentiation, LTP. It argues that the repetition of a task results in neuroplasticity. LTP also leads to dendritic branching or dendritic arborization, which leads to an increase in neural density. So more you are using your brain for a particular thing, more dendritic branching is happening. That means you are getting better in doing that ability. The neural density is increasing, you are doing better in that particular ability or skill. So lack of potentiation as a result of discontinuation behavior may lead to neural pruning, the process by which extra neurons and synaptic connections are eliminated. So for whatever thing you are not using your brain, those connections are going to break or going to shed. So that is the pruning. Now I made you understand in the layman language, but here I am using some technical terms because I want you to write these in your answers to get better marks. So make sure that you use this kind of terminologies in your answers. Now, to support this concept, it is very important for IB psychology students to use studies as an evidence. Now, we have Maguire study, very interesting. It was done on London taxi drivers. Have you ever sit in a taxi? And have you ever seen that taxi drivers, when you are telling them to go by the main road and if there is too much of traffic there, they just, turn their wheel and steering wheel and they can take the inside roads, the peripheral lanes, and they can still get you out of the traffic jam. So how do they really know all this map of the city? It's like on the back of their hand, isn't it? Their spatial ability is amazingly well. So we want to know biologically, is the brain of a taxi driver any different from the brains of other normal people? So what is the biological difference here? So this study is of Maguire 2000, a very interesting study. This was to see that the London taxi driver's brain was any different from the other common people's brain. And in this study, there were around 16 right-handed males London taxi drivers, and they were all compared with the MRI scans of 50 right-handed males who did not drive taxis. So there are two experimental conditions. So there are two conditions here. One is experimental group, in where we have 15 taxi drivers, all are right-handed males, and on the other hand, in the control group, we have non-taxi drivers, which are 50 males. And in order to take part in the study, the participants had to give the knowledge test and a license that would say that, they had license for over 1.5 years minimum, and the MRI database was collected. So the sample included a range of ages, so there was no confounding variable at all. The study is correlational in nature because there was no IV as such manipulated by the researcher, and the researcher was looking that if there is a relationship between the number of years of driving taxi and the anatomy of one's brain. So it was a single blind study that the researcher did not know whether she was looking at the scan of a driver or a control. It was a single blind study. The researcher did not know whether she is looking at the MRI scan of the driver or of a non-driver. So that was a single blind study to remove any research biasness. The data from the MRI was measured using two different techniques: voxel-based morphometry, that is VBM, and pixel counting. So what is voxel-based morphology? It is used to study to measure the density of gray matter in the brain, and pixel counting consists the counting of the pixel in the images provided by MRI scans in order to calculate the area of the hippocampus. So one is the gray area of your brain, and the other is the hippocampus, the area of the hippocampus that was studied in this study. Now there are two main key findings of this study. So the pixel counting here showed in an MRI that the the upper part, posterior part of hippocampus was larger in taxi drivers than the non-taxi drivers, and the anterior part of hippocampus was less in the taxi drivers as compared to the non-taxi drivers. So what exactly does this mean? And the VBM showed that the volume of right posterior hippocampus correlated with the amount of time spent as a taxi driver. So the volume of right posterior hippocampus was larger or was correlated with since how long they have been into driving the taxi. So what exactly does it mean? It means that posterior hippocampus is involved when previously learned spatial information is used, whereas the anterior hippocampal region may be more involved during the encoding of new environmental layouts. Let's do the evaluation of this study, and this clearly shows that there is definitely the difference between the brain structure of a taxi driver if you compare it with a non-taxi driver. So the evaluation of the study was, it was a quasi-experiment. That means there is no cause and no effect in the relation, no effect, cause and effect relationship can be established. So there is no independent or dependent variable, it was naturally occurring. The brain scans were coded so that analysis could be done blindly. The researchers did not know that they were looking at the MRI scans of whom. Is it the driver or the non-driver? So it avoids any kind of researcher biasness. Since this is a correlational study, it is difficult to say what is the cause and what is the effect. Is it that because they were taxi drivers, that is why their posterior hippocampus was large? Or it could be the other way around. Because the posterior hippocampus was large, they decided to become a taxi driver. Like for example, if you have a good skill in something, if my posterior hippocampus is big and I'm very good in understanding the spatial ability, I'm very high in spatial ability, I understand the road maps and everything very quickly, maybe I decide to become a taxi driver because I'm good at it. Then another is that it appears to have a sampling bias because all were from London, all were right-handed, all were males. So this is a kind of a biased sample that we have in the study. There are no females, there are no non-London, there are no left-handed drivers in this. That means this study cannot be generalized to a larger population but to a very specific kind of a population. And ethically sound it is because MRI is done, and it does MRI, any kind of biological test always gives a very objective result and very trustworthy, reliable result. It is not subjective at all. So MRI was done and it is not at all harmful. So it is a very good ethically used test. There is this second study, Dragansky et al. In 2004. Even this is a very interesting study of a juggler. So let's see what is there in the study. So the aim of the study was to see whether learning a new skill, in this case we are talking about juggling, can have any effect on the brains of the participant. So in this, the participants in this study, the participants were 24 volunteers between the age of 20 and 24. And there were 21 females and 3 males, and all the participants were non-jugglers at the start of the study. Each participant had an MRI scan at the start of the study. Participants were allocated to one of two conditions. So either the jugglers or the non-jugglers. Now those who were in the experimental condition, that means the juggling condition, they were taught how to use 3 balls cascade to juggle, like for a juggling routine. And they were asked to practice this routine and to notify the researchers when they had mastered it. At that point, the jugglers had a second MRI scan. After the scan, they were told not to juggle anymore, and then a third and final scan was carried out 3 months later. Now in the control group, the non-juggling group served as a control group for the duration of the study. To analyze the MRI scans, the researcher used voxel-based morphometry (VBM) to determine if there were any significant differences in the neural density gray matter in the brains of jugglers versus non-jugglers. So from the baseline scans, they found that no significant regional differences in grey matter between the two conditions. But the jugglers showed a significantly large amount of grey matter in the mid-temporal area of both hemispheres, an area associated with visual memory. Now, 3 months after the participants stopped juggling and many were no longer able to carry out the routine, the amount of grey matter in these parts of the brain decreased. There was no change over the duration of the study in the non-juggling sample. Now, very interestingly, it appears that juggling relies more on visual memory, that is the perception and spatial anticipation of moving objects, than on procedural or motor memories that would more likely show change in the cerebellum or basal ganglia. So basically, juggling is not about motor movements. So basically, juggling is not about procedural or motor memory. It is more about visual memory. So you are seeing things and the spatial ability, where the ball is going and then you kind of catch it. So this is more of spatial ability and visual memory, which is important in juggling. That is why this specific area of the brain started to have more gray matter. Now here is the diagram that you can see. So sample, there were two, jugglers and non-jugglers. Jugglers were practicing, non-jugglers were not practicing throughout the study. They were just a control group doing nothing at all. And there were 3 brain scans done for the jugglers initially before the study started, after 3 months, and then they stopped practicing, and then after 6 months. So basically what has happened here, I'll tell you, when the jugglers were there, initially the brain scans were showing that there was less gray area in this particular brain part. That less gray area was in mid-temporal brain part, mid-temporal area of the brain. And after they started doing juggling, this particular area, which is responsible for localization, visual memory, so this particular area which is responsible for visual memory, there the amount of gray matter had increased because the jugglers were using this specific part of the brain for juggling, because they were juggling and they were using this specific part of the brain. Hence the brain started to develop new neural connections in that particular area. But once they left it, after that they were not doing juggling anymore. What happened? The brain got a signal that this area now is not being used as much as it was being used before. So since this area was not being used anymore after 3 months, the connection started to break. So this is a very clear-cut study of seeing that how connections can be made when you start a new skill, learn a new skill, or you're doing something that is productive or creative or anything like that, and how connections start to break when you don't use your brain at all. So let's do the evaluation of the study. It was a pre-test, post-test design to show differences in neural density over time, which is a very good thing, because when there is a pre-test and there is a post-test, we are able to compare the results of the test. We are able to compare the results of the participants and see what are the differences. The differences give us a very good idea that some changes have been taken place. The second is the study was experimental, thus helping to argue for a cause and effect relationship. So we know that juggling is leading to synaptic connection. So there is a cause, there is a effect. IV and the DV is there. So it's an experimental study. Third was there was a control group that didn't juggle, that served as a control group. Now this was also very important because if we would have kept only experimental group, we could have seen their MRIs and see the differences. But then it was a good idea to keep another control group not doing anything at all and compare this experimental group with the control group. So this comparison gave us a better idea of the study. Then a sample size was very small. Now this is a weakness because the sample size was very small and the data may not be very reliable or generalizable to a larger population. Another problem was it had with internal validity, because participants were in their home environment for a good part of the study. Because they were in the home environment, we don't know how much they practiced. Did they practice or did they not practice? Or after 3 months, for the, when we told them to stop, whether they stopped practicing or they did not. So it was difficult to control a lot of extraneous variables. And last is the study would need to be replicated to establish its reliability, because, and there were 21 females as less number of males. Even that is an imbalance in the gender. That is again a problem of the sampling. And this study has not been replicated. So we need a replication of the study to confirm its results. So this was Dragansky, was a very interesting study to be written in neuroplasticity. There is another third study that I'm putting up here, an extra one, just in case you want to write more studies in your answer to get better marks. This is Mindfulness and Neuroplasticity by Holzel et al. 2011. In this, Lazar and Holzel have worked with research teams with the aim of investigating the physiological impact of mindfulness-based stress reduction programs on our brains. Now, this study consisted of 16 adult meditation-naive individuals whose whole brains were scanned using MRI technology. So basically, these were people who had never done meditation before in their life, and before the starting of the study, their brain was scanned using MRI. The participants then engaged in an 8-week mindfulness-based stress reduction program, MBSR. At the conclusion of the program, the brains were scanned again. The pre and post MBSR intervention scans were then compared using a technique called voxel-based morphometry. This technique analyzes MRI scans to detect physiological differences in gray matter. These results were then compared to a control group of 17 participants who were waitlisted for MBSR program. Now, the results were that participation in this program is associated with changes in gray matter concentration in brain regions involved with learning and memory processes, emotion regulation, self-referential processing, and perspective taking. So specifically, what is happening, the brain region of these participants in the experimental conditions who got into this program, in which the gray matter, neurons and dendrites increased in density, included the hippocampus, cerebellum, and cingulate cortex. You can see a graph here. So the controls who had not been in this program, the levels are pretty much low and in the gray matter area. But the ones who were in this pink column, in the pink bar, who were in the program, there was an increased density of gray matter in their hippocampus, their cerebellum, and cingulate cortex, which are basically used during the program of meditation, by doing the activity of meditation. Then we have a fourth study. This is not for the SL students, this is for the HL IB psychology students, because HL students might have to write one animal study. So one animal study for the HL students. This is remapping of sensory cortex by Merznick et al. 1984. So neuroplasticity on the level of cortical remapping. Now, researchers studied the cortical representation of the hand in 8 adult owl monkeys. There were 3 steps there. The first is sensory inputs from all the hand digits, fingers, were mapped in the cortex. To do this, electrodes were inserted in the cortical area known to be responsible for sensation from the hand. Then researchers stimulated various areas on all the fingers one by one and noted which electrode was responsible to the stimulation. Monkeys were given anesthesia before this procedure. Second step, the third digit, the middle finger of the monkey's hand, was amputated. I know that's very sad, but yes, that was done for the study. Now, 62 days later, a remapping was done to see how cortical area responsible for sensitivity from the hand changed after amputation. Now, what happened was, when the middle finger was amputated, results of the first mapping showed that there were 5 distinct areas in the brain, each responsible for one finger. And adjacent fingers were represented in the adjacent areas in the cortex. So what happens to the area responsible for the third finger after the finger is amputated? it was found that adjacent areas, those responsible for sensitivity from digit 2 and 4, spread and occupied parts of the row, now unused areas. The areas responsible for digits 2 and 4 became larger, while the areas responsible for digits 1 and 5 stayed the same. It was concluded that cortical remapping of sensory inputs from the hand occurred within 62 days in owl monkeys. The brain is fixed and certain psychological functions are hardwired in certain parts of the brain, but it can adapt itself to the demands of the environment. So the other senses may be used to make up for the lost sense is known as sense substitution. Now in this study, let's say the 5 fingers are there and each finger was seen that which area in the brain it is connected with. Let's say 1, 2, 3, 4 and 5. This is just an example, okay? Now here what happens, this area got amputated. Now what happens after 62 days, this number 2 and number 4, these areas where it was actually connected to this part of the brain, here the connections had widened and taken this part because now this finger doesn't exist anymore. So it took 62 days in an owl monkey for them to do the cortical remapping, and this area was covered by the number 2 and number 4 finger. So that is what it has been told in the study, that like I gave an example sometime back about a blind person. When some limb or some eyes or any sensation, we lose anything in our body, the other parts of the body try to make up for that very part, and that is called cortical remapping. So here we are done with neuroplasticity. I hope you enjoyed. It's a beautiful concept, and the takeaway is, the application takeaway here is that in your life, never think you are too old to learn something. There may think that now I'm very old, I'm growing old, how can I learn a new thing? So here we are done with the neuroplasticity. Extremely beautiful chapter, extremely beautiful concept it is. It clearly gives us hope and encouragement that it's never too late to learn something new. Of course, a child can learn faster than you because the being made very fast in the in a smaller child as compared to the older person, but still that does not mean that it's too late for you and you'll not be able to learn. You definitely can learn new things in life, and keep using your brain as much as possible to build up those synaptic connections strong enough